Hi, let's know about dyes used in electrophoresis techniques. Electrophoresis is used for separation of DNA, RNA, and proteins. To visualize or monitor the molecules during and after electrophoresis, dyes are used. Broadly, these are of two main types. 1. Staining dyes, used after or sometimes during electrophoresis to make the biomolecules visible. And second, tracking dyes, which are added before electrophoresis to monitor the migration of molecules and track progress. 1. Staining dyes in electrophoresis. Staining dyes bind specifically to biomolecules and allow visualization after electrophoresis. Are A. For nucleic acids, DNA and RNA, such as Ethidium Bromide, EtBr. Principle: Ethidium Bromide intercalates between bases of nucleic acids and fluoresces orange under UV light. Pros: it is sensitive, cheap, widely used and works with both DNA and RNA. Cons: it is mutagenic and carcinogenic, requires UV exposure, damaging to DNA, so less safe. Next, SYBR Green, SYBR Gold. Principle: it is a fluorescent dyes that bind nucleic acids. It is a safer alternative to EtBr. Pros: it is more sensitive than EtBr, less toxic, safer to handle, works in real-time PCR too. Cons: it is expensive, photobleaching occurs with prolonged UV exposure. Next is GelRed, GelGreen. Principle: these are large molecule dyes that intercalate into DNA but are designed to be non-toxic and impermeable to cells. Pros: it is also a safer alternative to EtBr, highly sensitive and stable. Cons: more costly than EtBr. Next is methylene blue, crystal violet. Principle: it stains nucleic acids by binding electrostatically. It is visible under normal light, no UV needed. Pros: it is safer, cheap, and non-carcinogenic. Cons: it has low sensitivity, requires high DNA concentration, may cause background staining. Now let us see dyes for proteins. 1. Coomassie Brilliant Blue, CBB. Principle: it binds to amino acid residues, mainly arginine aromatic residues in proteins. It is used in SDS page. Pros: it is easy to use, inexpensive, fairly sensitive, can detect approximately 0.1 to 1 microgram of protein. Cons: it is less sensitive than silver staining and requires destaining step. Next is silver staining. Principle: in this method, proteins reduce silver ions to metallic silver, producing dark bands. Pros: it is very sensitive, can detect nanogram levels of protein. Cons: it is labor-intensive, expensive, can produce variable results, not always compatible with downstream mass spectrometry. Next is fluorescent dyes. SYPRO Ruby, deep purple. Principle: it binds proteins and fluoresce under UV or laser scanners. Pros: it has high sensitivity, wide dynamic range, good for proteomics. Cons: requires fluorescence detection equipment, so costly. Next is Ponceau S stain. Principle: it is a reversible stain for proteins on membranes after Western blot transfer. Pros: it is quick, reversible, good for checking protein transfer. Cons: it is not permanent, low sensitivity, not good for quantitative work. Now about tracking dyes in electrophoresis. Tracking dyes are loading dyes mixed with the sample before electrophoresis. They help visualize migration during electrophoresis, estimate progress of the run, provide density to the sample, usually glycerol or sucrose is included. Common tracking dyes are 1. Bromophenol Blue, BPB. Migrates at a rate similar to small DNA fragments approx. 300 base pair length in agarose gel. Pros: it is inexpensive, stable, widely used, visible. Cons: it is not precise for larger fragments, may overlap with small DNA bands. Next is Xylene Cyanol. Migrates slower than BPB, approximately 4 to 5 kilo base pair of DNA size in agarose gel. Pros: useful for larger fragments, visible. Cons: sometimes faint, not as sharp as BPB. Next, Orange G. runs faster than BPB, approx 50 base pair of DNA size in agarose gel. Pros: it helps track very small DNA fragments. Cons: it is not widely used, faint visibility. Next is Cresol Red. Migration depends on buffer pH, often runs with medium-sized fragments, approx 125 base pair of DNA size in agarose. Pros: provides an additional marker option, visible in colored gels. Cons: it is less common, can be affected by pH changes. Thanks for watching. Please don't forget to like, share and subscribe the channel.